Hi guys, we're back again, and today we are going to talk about prokaryotic cells. But first, when we think of prokaryotic cells, what does the word pro remind you of? Pro means before. And karyon means nucleus. So here is a cell, very primitive cell, and that must have existed without a true well-formed nucleus. So if you see a prokaryotic cell, there are certain parts that you will first see. First and foremost, there are these extracellular appendages. Well, the first appendage that we are looking at is, of course, this long whip-like structure which is made up of a protein. This is the long whip-like flagellum. Some bacteria have this, some bacteria do not have this, and of course the function is movement. Second, they have these small strand-like structures called the pili, and the functions of the pili, which are shorter than the flagella, of course, but the function is different. The function is mostly attachment, so here is attachment. And the attachment to surfaces, also like twitching movement. So remember, this is just form of a twitch. You can twitch. And gliding movement. But some pili are sex pili. And these are used for sexual reproduction, where they are going to exchange genetic material from one cell to another. The surface layers. Well, the surface layers are the outermost layer, the glycocalyx. The glycocalyx is a thin gel-like layer which surrounds this. It could be a slime layer. The slime layer is very irregular. It is very diffuse. And then there's a firmer layer, which is the capsule, which is more distinct and more firm, more gelatinous. The function of the slime or the capsule layer is the same. Well, of course, your favorite word, the function of this, I'm going to write it out here because I'm out of space, is protection. And so protection and, of course, attachment. And remember, this is attachment to different types of surfaces. A bacteria needs to bind to a surface, like, for example, plaque on your teeth. Protection would be, it will protect the bacterial cell from immune response. And this is how some bacteria escape immune detection. Or also they need, it can provide protection from environmental factors. The second one is the cell wall. So here's the cell wall. The cell wall in a bacteria is unique. The cell wall is made up of peptidoglycan. So unique. This is DO. So peptidoglycan. And here the cell wall, of course, is going to distinct the bacteria into two types. You don't need to know that, the gram-positive and the gram-negative. But it doesn't matter. All cell walls in bacteria are made up of this unique mucopolysaccharide called peptidoglycan. The inner layer is, of course, the cell membrane. So here's a plasma membrane or the cell membrane. The cell membrane, like eukaryotic cell, is also made up of bilayer of phospholipid. So here is two layers of phospholipid, as you can see. This is 1 and 2. And in between, you have the proteins that are floating, that are going to be floating around in a fluid mosaic model. So this is exactly like the cell membrane we've talked about before, the biphospholipid layer. And what is the function of the cell membrane? The cell membrane is selectively permeable. It will only allow certain substances to pass through. Okay. Then, so we have the glycocalyx, cell wall, and cell membrane done. And then you have in the intracellular spaces, you have a cytoplasm. Now, cytoplasm is this living jelly. This living liquid jelly is the cytoplasm, which provides the living environment to the inner structures of the bacteria. Cytoskeleton, for the longest time, scientists thought that there's no cytoskeleton in a prokaryotic cell. But now they're finding out that there is cytoskeleton, and just like eukaryotes, not as advanced as that of eukaryotes, but definitely there's a cytoskeleton which forms like a mesh, holds everything in place and gives support inside the nucleus, inside the bacterial cell. Ribosome. The ribosomes, they do have ribosomes, but this time the ribosomes are going to be much smaller than eukaryotic. And not that you need to memorize, but this is only 70S as opposed to a eukaryotic ribosome, which is a much bigger unit. Now here the ribosomes are scattered in the cytoplasm or remain attached to the cell membrane. So these are the ribosomes. The function of ribosome, like in eukaryotic, is of course protein synthesis. There are storage granules, of course. There are these food granules, which are just inclusions. They are not living part of the cell. There's a nucleoid region. Remember, the nucleoid region is not the same as nucleus. Why? Because there's no nuclear membrane. So you need to notice how simple a prokaryotic cell is. There's no nuclear membrane. There's single circular DNA. So there's a single circular chromosome. And of course, this chromosome is going to be made up of DNA. So the single circular chromosome is found just floating around here in the cytoplasm. The region around it is called nucleoid region, which of course you understand is not the same thing as having a well-defined nucleus. Another structure which is not drawn here are the plasmids. The plasmids are small circular loops of DNA. Okay? But the difference between this plasmid and this single circular chromosome is that these plasmids are definitely going to carry very few genes. Now, the bacteria can have many plasmids, and what these plasmids provide some kind of genetic advantage to the bacteria. For example, antibiotic resistance, which is now a big problem, as you know, with bacteria. So when the bacterium divides, then all of the plasmids contained are also copied such that each daughter cell receives a copy of the plasmid. The bacteria also can exchange plasmids amongst themselves. So that is the structure of a prokaryotic cell. Now, if you compare the differences between prokaryotic and eukaryotic cell, you will notice that a DNA or a chromosome in a eukaryotic are linear strands within membrane-bound nucleus, the nuclear membrane. Now, there's a single circular chromosome in a region which is called the nucleoid region. Size. The eukaryotic are way larger than a prokaryotic cell, almost 10 times larger than a prokaryotic cell. Organization. Well, eukaryotics are mostly multicellular. Some are, of course, unicellular too, but often they are multicellular, and some have cell walls. Like plants, they have cell wall, or fungi, they have chitin. But the cell wall is not made up of peptidoglycan. Bacteria are all single-celled. They have a very unique cell wall made up of peptidoglycan. Metabolism, of course, most of the eukaryotes need oxygen. Prokaryotes may or may not need oxygen. They have a very unique type of metabolism and respiration, and that is called anaerobic. That means they do not need oxygen. Okay? Organelles, they are membrane-bound organelles like mitochondria, Golgi, ER, chloroplast. Well, there's no membrane-bound. It should not say organelle, but no membrane-bound organelles like, so here of course we are talking about ER or Golgi. Now here also another difference is that the ribosome out here is different from the ribosome in eukaryote. Now eukaryote, the ribosomes are large, so they have larger ribosomes. Here they have smaller ribosomes. Examples of course will be plants, animals, protists, fungi. Here they are bacteria as well as archaea. So I guess with this we are come to the end of our presentation. If there are any questions or concerns, please stop by my office, call me or talk to me. Thank you for giving me your attention and time.